Hello and welcome to another edition of Lost in Science. This week we are taking a little break from new stories, but we do have a special guest on the show. We have Spider-Man. Well, a Spider-Man. Claire is talking to Joseph Schubert about peacock spiders and a newly discovered species. Well, it was newly discovered. This is a bit of an old story from the archives. And also, if you've ever wondered what the answer to the old riddle, my shark's got no nose, how does he smell? We'll have to ask Claire to ask an expert. Claire is talking to Victoria Camilleri Asch about how do sharks actually smell underwater. So please stay tuned for these archival stories from Lost in Science.
We love a citizen science story on Lost in Science, especially when it leads to the discovery of new species. My guest today is an arachnologist, here to tell us about his latest described species of the incredibly charismatic group of spiders, the peacock spiders, with the help of citizen scientists. Joseph Schubert, arachnologist from Melbourne Museum. Welcome to Lost in Science.
Thank you for having me.
Okay, so Joseph, first things first, can you describe peacock spiders?
Okay, so if I were to describe a peacock spider, I would like you to imagine a tiny 4mm 8-legged kitten. They're tiny little jumping spiders which are incredibly colourful, and they love to dance. So these spiders perform courtship displays. The males are really colourful and they'll display their colours to females that they're trying to mate with.
Did you say an 8-legged kitten?
Yes, they're really cute, I find. And yeah, they've been compared to kittens before.
What do you think about them makes them so cute?
Well, they've got these massive forward-facing eyes, which I guess makes them more relatable to arachnophobes. And they're really, really tiny, so they're completely harmless and they just hop around and they're very, very curious animals.
Oh, they sound adorable, like sort of like a nice entry into the world of spiders for a lot of people, maybe?
Yeah, definitely. A lot of people who are arachnophobic have told me, oh, like, I'm scared of spiders, but these are actually kind of cute, which I love to hear.
That's great. That's excellent. Is it only the males that have the colourful display?
Yeah, that's right. So only the males are colourful, and actually only when they're mature. So they spend most of their lives brown in colour, and they go through a process called molting, which is where they shed their skin. And each time they shed their skin, they get a little bit bigger. And in their final molt, which is when they're mature, their colours and their reproductive organs will develop.
Oh, right. And are we talking like specific colours on the spectrum, like greens and blues, like the peacock, or is it more diverse than that?
Yeah, they're massively diverse. So some are greens and blues, some are reds. And then in this case, we've got an orange one. So they're a massive range of colours.
How common are they in Australia? Is there a good chance that, I might have seen one or passed one without even knowing?
Yeah, they're incredibly easily overlooked, but they're very, very common. So in most natural habitats, they're able to be found on the ground usually, or near the ground, usually in leaf litter or on twigs or on grass. I've actually found them in my backyard in suburban Melbourne, so they're quite common indeed.
Whoa, suburban as well.
Yes.
So they occupy a lot of different places.
Yeah, that's right. And in a variety of habitats too. So they range from coastal habitats to the desert. And then in this case, with this new species, to an ephemeral swampland, which is really amazing.
Okay, so you've alluded to a new species a couple of times. I'm very excited to talk to you about that. Can you tell us a bit about this new species that's just been published?
Yeah, so this new species is an orange and white-faced cute little peacock spider, which I've decided to call Maratus nemo. named after the clownfish. And this species was actually discovered by a citizen scientist who was doing some ecological field work. She's actually professional with plants, but she happened to be taking photos of jumping spiders because she's interested in them as well, and posted them on a Facebook group. I came across her post and thought, wow, that looks like a new species. And here we are.
This is the first time this particular Nemo spider has ever been described.
Yeah, so I'd never even seen photographs of it before, which is really amazing. And it seems to occupy quite a short range in a very particular type of habitat. So it's unsurprising that no one's really looked very closely at that type of habitat before for these jumping spiders. And yeah, it's really amazing that she was the first one to photograph it and post it.
Wow. Do you have to go out into that habitat and find other specimens of that particular peacock spider to verify that it's a new species? What's that process like?
Yeah, so typically I would go out into the field and collect specimens myself, but Cheryl, who found it, was actually kind enough to collect me some specimens, which I was able to then examine in the lab, which was really cool.
Yeah, that's awesome. And do you think it's only localized to the place where Cheryl found it, or what are your thoughts around, how it's distributed?
Well, it's difficult to say. So it probably could occupy a larger range than we currently know. There are some species which we only knew to occur in Western Australia that we now know occur in Victoria. So ranges can be massively extended. So it's just a matter of looking more to find out.
And I understand that the process of naming a new species is not an easy one. Has this been a long time coming?
Yeah, it's been quite a few months in the making. So she collected the specimens in November last year, and the paper was only just published a few days ago. So quite a long time.
Joseph, how important do you think citizen science is in describing and protecting biodiversity?
I think it's massively important. Realistically, the scientists who specialize on groups like these can't be everywhere at once. So to have thousands of pairs of eyes looking for new species is incredibly helpful. And also a lot of the time we're stuck in the lab and don't get to do as much field work as we like. So having more people out there looking is really, really good.
And how would you recommend people get involved with citizen science projects?
Well, I think just getting out there and taking photographs of interesting looking critters and recording the locality data and uploading them to websites like iNaturalist or onto Facebook groups, I think that's a very, very helpful first step. And then if you have found something interesting, we can use the photographs and the locality data to expand further on a potential discovery.
So, I mean, it's interesting to hear, you as an arachnologist are scouring the Facebook pages for, new photos of things. Would that be a common sort of thing for scientists across the board and biologists across the board to be doing?
Yeah, it's definitely becoming a lot more common. I have seen a number of professional taxonomists on a number of these different Facebook groups, even for different taxonomic groups, not just spiders. So yeah, people are definitely using social media to science's advantage.
That is a outcome of social media that I wasn't expecting.
Yeah, it can be used for good sometimes.
So Joseph, how many peacock spiders in Australia have been described?
With this edition, it makes it 92 species, so quite a few.
That's a massive amount. Do you think there are more out there?
I'm quite sure there would be. So 77 or 78 or so of these 92 species have been described and discovered in the last 10 years alone. So it would be completely unsurprising for more to be out there. In fact, I've definitely seen photographs of what are likely some undocumented species on various platforms like iNaturalist and on social media. So it's just a matter of getting out there and collecting specimens for scientists to do the taxonomic work on them.
You're a peacock spider detective. You've got a couple of leads in the field.
That's what it feels like sometimes, doing a bit of detective work, and I think that's what makes it really fun.
Speaking of field work and having fun, are there any hilarious stories from the field or working with peacock spiders?
Probably one of my most memorable stories in the field was working with a mentor of mine named Barbara Bear. She's amazing. She's described hundreds and hundreds of species. And we were out together, and I was putting a wolf spider into a jar, and it ran up my arm, and I honestly freaked out, and I screamed, and I flicked it off me, and she was laughing at me, and she was like, "Oh, I thought you were a brave arachnologist, Joseph. What's going on here?" And anyway, we had our laugh, and then a few minutes later, the exact same thing happened to her. It ran up her arm, and she let out a big scream, and then I was like, there we go, Barbara. Who's laughing now?
That is excellent to know. I'm sure, our listeners around Australia will be very happy to know that even arachnologists get freaked out when spiders run up your arm.
Yeah, so I'm a little bit of an arachnophobe, but I still love them all and appreciate them.
Excellent. Well, Joseph, a big thank you for coming on Lost in Science today and sharing your spidery stories. Congratulations to you and Cheryl on the new species, Maritus nemo, if I got that right.
That's right, yep.
Yes. And best of luck with the hunt for new species of peacock spider. Hope to talk to you again soon.
Awesome. Thank you very much for having me on. See you soon.
It's a big scientific experiment. What do I know?
Across Australia on the Community Radio Network, you're listening to Lost in Science.
So I can't remember when I first heard that sharks could smell one drop of blood in a whole beach, but it certainly had me thinking twice about swimming in the oceans with cuts or abrasions. But what does science really know about shark smell and how it works? Are shark noses really as sensitive as we think? Well, our guest on Lost in Science this week is very qualified to give us an answer to confirm or deny my childhood facts. Dr. Victoria Camilleri Asch is a fish biologist and postdoctoral research fellow at Queensland University of Technology. Victoria, welcome to Lost in Science.
Thank you, Claire, for having me.
So, Victoria, do sharks smell as well as we think?
That's the very question that made me start my PhD. So the main thing, I guess, to consider is when you assess an animal's ability to detect some signals, let's say olfactory cues in our case, you can assess it looking at how the olfactory system is organized. That's morphology. You can assess it looking at how it functions, and that's physiology. Or you can look at how the behavioral outputs are displayed, and that's more ethology, so animal behavioral research. And now you can add another layer actually that I forgot is molecular biology. You can look at the genetics side of things. So depending on how many genes are expressed to actually do that task, you can also assess it this way. When I started my PhD research, I was interested in the holistic answer, right? Yes or no?
Yeah. Yes or no?
And I know. That's but when you dig down and you look at what's been done, what remains to be done, you kind of come up with an answer which is still in the gray area. Bluntly, there is no yes or no answers. as just yet. It just depends on what you look at. So if you look at the genetics, for instance, like for fun facts, humans have roughly 800 olfactory genes, primates 600, mice 1400.
Wow.
Rats even a bit more than that. Dogs 1100, so a bit less. Cows 2000.
Cows.
Yes, so far they're topping it. Other fish roughly 300 and something.
Okay.
And sharks, you go down to sharks, elephant sharks or other sharks, about 50 to 60 genes. So if you look at just the genetics, it doesn't seem like they have a lot of genetic information expressing receptors able, to collect that in kind of information. But then when you look at the brain and the size of the olfactory bulbs compared to relative to the overall brain size, then sharks have quite big olfactory bulbs. So the brain area used to process that kind of signals, so odors, is relatively large compared to other animals. So that's another way to look at it. So morphology.
Right. So that's in morphology. You got your genetics, and then you've got your physiology as well.
So in terms of physiology, for instance, the story is a bit different. The sharks can have detection thresholds, so the minimum amount of chemical that can be detected, quite low. So 1 part in a billion, for instance, for some of the chemicals out there, like amino acids, or some of them anyway. So research showed that bony fishes are able to detect as low detection threshold for pheromones, for instance, as feeding cues for sharks. So for sharks, we don't know for the pheromones how low that detection threshold might be. That's a research happening right now by a good colleague of mine at UQ. So we're going to find out probably soon. The question is not out there yet. So if you want to look into this, my good friend Heather Middleton is looking at this at UQ, and she's probably going to share that story when it's out there.
Okay, so it sounds like shark smell is a lot more complicated than we thought, but they do have that morphological, that sort of like enlarged parts of the brain, even though they might not have as many genes as other different animals. So when you went into your PhD, what was your question, and what were your sort of like research questions about shark smell?
Yeah, so based on the recap you just did, I guess the main thing that I stumbled upon, and I really wanted to get down to knowing more about that, was the morphology aspect. We seem to have done quite a lot of research on behavioral responses or physiological responses of some chemicals, but not necessarily how the system itself, the wiring to the brain or within the brain, was organized compared to other animals, which can answer one bit of the question, if you like, and hasn't been questioned yet as much. So that's the very question I tackled, and I wish I had time to tackle more, and I didn't in the end. So I stuck to anatomy pretty much.
And I guess with shark anatomy, you need to go and have a look at some sharks and look at their brains. So
absolutely,
take us through how you went about that.
That's a very good point, actually. So working on these animals is not necessarily easy because, you need to access some samples or them in nature. So I was lucky, because I chose a lab, the neuroecology group, which was led by Professor Sean Cullen at UWA. When I started, he had a huge collection of shark samples in his lab, amongst other animals, being a sensory biology lab. And so my other co-supervisor, Dr. Kara Yopak, or Professor Shark Brain, if you want to call her this way.
Professor Shark Brain.
Yes, she's out there. She's based
excellent
in North Carolina, at the University of North Carolina, Wilmington now, but she was at UWA as well when I started, and she had a huge collection of shark brains over there. So I guess it was the best way to start, just working from sample off the shelves, instead of going out there and having to apply for permits and euthanize more animals that was needed.
And the actual sort of lab work that you then did, the comparative anatomy, or take us through what some of your experiments were like.
Yeah, so going for anatomy is one thing, but then what questions exactly? So there is a nice body of research that has used olfactory bulbs, so that part of the brain processing essentially olfactory information. So and this has been used as a proxy, or their size, their shape, or the volume has been used as a proxy for olfactory abilities across many animal taxa, including fishes. So that was the starting point, which was also a research area of my co-supervisor, Cara. She had extensively compared the size of these bulbs across many, many shark species and rays and even chimeras. And so the question was, okay, this has been accepted as one way of comparing across species, but do we know what it means? Because it's one thing to have a bigger brain structure, but if inside you don't have as many neurons because they're less densely packed, then maybe your processing power is not as great. And so the size might not reveal you what you actually want to test, you know? So that was one of the biggest questions we wanted to start with. So in trying to answer it, I came across a new technique to visualize brains. So MRI, same as for us humans, is one way to do it if you want to calculate the volume of a brain area. And it can be a very expensive technique if you have a lot of specimen to go through, and not as handy because, the training and the access to the hospital is a limitation, obviously. So we came across that new technique using CT. So it's the same as a CT scan, same as humans or akin veterinary science, same kind of apparatus, but quite for smaller samples, so really high resolution. But it had never been used, that imaging tool, to that application. So that was another thing. Let's try to use something new and see if we can get an answer, which we did.
Right. So and then what did you find?
So what we found is that, to put it in a nutshell, the volume of the olfactory bulb was assessed using that technique successfully. So that was one win. Then we compared the olfactory bulb size across, two main representative fish species. So one cartilaginous, a cat shark. And
right.
one,
so cartilaginous fish are fish like sharks they don't have bones. They have cartilage, right?
More or less, their skeleton is mainly cartilage compared to bony fishes. And when we say cartilaginous fishes, also just a precision for those that might be listening and not in that field of research, we mean shark, skates, and rays. They call the elasmobranchs, but there is also chimeras, and all of that are cartilaginous fishes. So
right.
Coming back to your question, the main finding is that, we use that novel imaging tool to get the volume of the bulbs in the two representative species. But then we used another microscopy technique to get the number of neurons entering the olfactory bulb and exiting. So we could actually test whether or not the volume was meaning something. And we did. We actually found that in the cartilaginous species, there was much more neurons entering the olfactory bulb, and they were packed the same as in the bony species, the bony fish species. So we could tell that based on how they are compacted or how dense they are, which was exactly the same between the two different species, it was just based on a difference of neuron entering that brain area that explained how the olfactory bulb was bigger.
So Victoria, as a shark biologist, what is one way, I guess, sharks cop a lot of flack for being man-eaters, but what is one thing that you want people to understand better about the nature of sharks?
The first answer that comes to my mind is part of your question, is just to better understand them and to seek answers for things you believe about them. They're not that well understood, and to be honest, even with the research conducted so far, we certainly know more. But even everything that I said to you today in our session, I can't answer if yes or no they are that good at smelling compared to other animals, So I think there is really a call to better understand them, or try to, and to better protect and conserve them then. Because there is no way you can tackle big question as sustainable human-animal interaction if you don't understand the subject, and that's both ways. So we have a lot of information for the human side of the story, but for the animal side of the story, there is a lot to answer. So generally understanding more about their ecology, or ways to reduce their captures in fisheries, which is their highest threat today, or just how they are the awesome animals that they evolved to be throughout million years. It's probably my only take-home message today.
Well, Dr. Victoria Camilleri Asch, fish biologist, thank you so much for coming on Lost in Science today. Thank you so much for sharing your passion and your research into shark biology. And I'm certainly going to think differently next time I see sharks either diving or at the aquarium. Absolutely. I'm going to wonder a little bit more about their brain morphology and what they can smell right now.
Thank you for having me again.
And that is all we have time for this week on Lost in Science. Thank you for joining us in Getting Lost. If you have any questions or suggestions for the team, get in touch with us by email. We are lostinsci@gmail.com. You can send cheap tweets to us at lostinscience1 on Twitter, or you can find us on the ubiquitous Facebook. Lost in Science is recorded at the studios of 3CR in Melbourne on the land of the Kulin Nation, and is broadcast across Australia on the Community Radio Network with the assistance of the Community Broadcasting Foundation. You can find a podcast version of the show on 3cr.org.au, or you can tune in the way you did this week when we return in our usual time slot to get Lost in Science!
Thanks for listening to a 3CR podcast. 3CR is an independent community radio station based in Melbourne, Australia. We need your financial support to keep going. Go to www.3cr.org.au for more information and to donate online.